This video is going to be very important. It's our introduction to the material covered in chapter 11 and 14. There's going to be a lot of key vocabulary that you need to absolutely understand that we're going to kind of build as our foundation. And we'll also look at the kind of cell division, the special kind of cell division that makes the gametes, which are the sperm or the egg. And that kind of cell division is not called mitosis, which we learned about in the last unit. It's called meiosis. As we looked at in the last unit, as we kind of mentioned briefly, there's this structure here, which is called a karyotype. And what a karyotype shows is all the chromosomes, which is that condensed DNA, for an organism. This happens to be the karyotype for a human. And in human beings, we have 46 chromosomes total. We have them in 23 matched pairs. So for our species, and this is not the case for all species, but for our species, we have 2 chromosomes for each of what we call a homologous pair. And we have 23 of those pairs. So 2 times 23 is a total of 46. Now, the 23rd pair is a special group. So this 23rd pair is pretty important. It's pretty special because they're called the sex chromosomes. And these chromosomes are what determines the sex or the gender of the person. So XY is a male. If it was 2 X chromosomes, it'd be a female. And then so the rest of these are what we call autosomal chromosomes. Now, a real quick side note here, it is actually the father who determines the sex of a child. And the reason for that, remember, women are XX. And so both of their eggs are going to have an X chromosome in. There's no other chromosome they can donate. But men are XY. So if you think about the sperm that are produced in a male, half of those sperm will be X sperm, and half of them will have the Y chromosome sperm. And so it depends on which sperm fertilizes the egg, whether you're going to have a baby girl or a baby boy. So it's technically the father that actually determines that. So a little side note there, and this will make a little more sense after we actually talk about, how are these eggs and sperm formed in meiosis. So we already know the 23rd pair is the sex chromosomes. All the rest of the chromosomes that are not sex chromosomes, these are called autosomal chromosomes, or just simply autosomes. So what pairs are we talking about? Obviously we're talking about pairs 1 through 22, because we know that the 23rd pair is our special sex chromosomes. So the rest of them, all the rest of them, these are our autosomal chromosomes. So there are 2 terms you definitely need to know for sure: sex chromosomes, autosomal chromosomes. So in addition to autosome and sex chromosomes being important definitions, there's 2 other words that you need to know, and that's called somatic cells and gametes. Very simple. Gametes are the special cells that are used for sexual reproduction, which, as I showed in the last slide, that's the sperm and egg, right? Depending on if you're a male or female making sperm or egg. All the rest of a human being's body is built from a different kind of cell called a somatic cell. Now, in human beings, our somatic cells happen to be diploid. Here's another term we need to know: diploid. So just think about what this word means. Di means what? 2. What does the ploid mean? Ploidy essentially means how many chromosomes do we have for each homologous pair. So humans are diploid, so we are 2N. If you think back, if you look at that karyotype in front of you, what you'll see is that for each of the homologous pairs, all 23, you have 2 chromosomes, so we are diploid. If we had, imagine, 3 chromosomes for each of those, we'd be triploid. If you had 4, quadruploid, or tetraploid, and so forth, like pentaploid, etc. Now, for humans, because again we have 23 matched pairs, our total number of chromosomes then is 46. 2 times 23 is 46. So realize all of our somatic cells, whether you're talking about, the skin, a heart cell, a liver cell, a pancreas cell, all of those cells are going to have a nucleus in them that's going to have 46 chromosomes. But the gametes that are used for sexual reproduction, they're not going to have 46 chromosomes. Gametes have exactly half as much DNA. They are what we call haploid. They are not 2N, they are N. Now that means that in our gametes, you've got 1 of each of the chromosomes, okay? And so that's important then, because then when those 2 gametes come together, when the sperm and the egg come together, they're each bringing 1 chromosome for each of the 23 pairs. So then what is the offspring going to have? When the fertilization happens, they'll have 46 again. So this is kind of the best way I can explain it. This is the visual. So here is a human karyotype, by the way, male or female. Well, pretty easy for you to know. You go to the 23rd pair, we see an X and an X, clearly a female. If you count the chromosomes, you'll find that there are 23 pairs. So 23 times 2 is 46. There are 46 total chromosomes. But if you think about, okay, this woman, because we know it's a woman here, This woman, where did her chromosomes come from? Well, she came, like all of us, from a sperm and an egg, and each of those gametes, that sperm and egg, was haploid. It wasn't diploid like the last picture. It's haploid. It has one chromosome for each of the 23 pairs. But when the sperm that's haploid meets the egg that's haploid, you get one chromosome for each pair from each. And so now the zygote, that woman that's been created, she got one X chromosome from her dad, the other X chromosome from her mom. She got one chromosome number 1 from dad, she got one chromosome from mom, and so now she's got two chromosome number 1s, and so forth. So this is how it works. You go from, in humans, haploid egg and sperm. When they fertilize, you get then the 2N individual, the diploid individual. So, a little side note, what kind of microscope took that picture? I'm hoping you realize that it clearly must be an electron microscope because it's over 1,000x, it's 80,000 according to the picture. And it's in 3D, so that's a scanning electron microscope. So we know in human beings, that's what happens. You get haploid sperm, haploid egg, make a diploid individual, and then there's that diploid adult that's going to go on to make sperm or egg again, depending. This is a woman, so she'll be making eggs. It'll be haploid, and when she has a child, and so forth, it'll just go round and round. But this is not the case for all organisms. So let me show you a different karyotype here. So this is an individual, this is a different species. We don't have to worry about what species it is specifically. But this species is not diploid, as you can tell. A diploid individual would have, if you think about it, a diploid individual would have just these chromosomes, right? The first two from each pair, all right? But that's not the case here. Here we have, instead, we have 8 chromosomes for each pair. So this individual is not diploid or even triploid or tetraploid. This individual is octoploid. They have 8 chromosomes for each pair. So that's what this particular species is. And there are some species like this. For example, strawberry plants are octoploid. Now, this is not a strawberry plant's karyotype, because I just made this one up, but this is what it kind of would look like. So you've got 8 chromosomes, or pairs of chromosomes for each of the 23, in this case, pairs. Now, what would their gametes look like? If the individual, the adult, is octoploid, how many chromosomes are going to be in the gametes? Hopefully realizing that they'd be tetraploid, 4 in each. So that when they come together, you then get the octoploid gamete. So this is the way it works. This should not be too difficult for you, hopefully, to understand. On the exam, I will give you both directions. I'll say, hey, in a rose, let's say, the sperm and egg, the gametes, are triploid. So how many chromosomes are in the adult? And you just know, okay, 3N is going to be 6N. And then I'll do it the other way around as well. I'll say, okay, and let's say, some other plant, they have 120 chromosomes in the adult plant. Well, then they're going to have 60 in the sperm and the egg. They're going to have half of that. So be ready for those kinds of questions on the exam. With that under our belt, let's take a look at this picture here, and you're missing some labels, so let's make sure we label it here. So, in humans, we're back talking about humans now. Humans, you have a haploid 23 chromosome sperm, haploid 23 chromosome egg. When you put them together, make a little note, this term right here, this is called fertilization. When fertilization occurs, you then get your diploid zygote that has 2N chromosomes, so it's diploid, total of 46. One of those, one chromosome for each of the 23 pairs comes from sperm, and the other one comes from egg. Please also label this picture. This picture shows generally how it works. We've got, in humans, again we're talking about humans, a diploid female, diploid male. They're each going to go through meiosis. Circle, star, or highlight these terms right here. There's a special kind of cell division we'll look at in a minute that produces these gametes. And what's cool about this cell division is that you end up with daughter cells that end up with half as much DNA as the original mother cell, which is why they've gone from being 2N to now N haploid. And then in fertilization, so this is in the sort of the reproduction stage now, sperm meets egg, and they're each N, and so they're going to be now the zygote that is 2N, right? And then that is going to become the adult, either the adult male or the adult female, and around this cycle goes. This is how sexual reproduction works. So in the picture, you might want to make a little note over here. In the picture, we have a man and a woman, a human. But you could just as easily have, 2 dogs here, or 2 cats, or 2 elephants, or even two fish or even two plants. Anything that does sexual reproduction is going to have a cycle very similar to this, where you're going to go cycling between a 2N individual and then an N individual, or an 8N individual, like I said in the case of strawberry plants, and a 4N gametes, right? So around and around it goes. This is how sexual reproduction works in general. Fertilization is this special name for what happens when sperm meets egg, and the result of fertilization then is what we call a zygote. So this is that one cell that we all started life as. It's got its own kind of special name. It's called a zygote. Make sure you know those two terms as well. Now I want to take just a minute and I want to make sure that we're clear on the difference between homologous chromosomes, sister chromatids, and even genes and all these kinds of things. So human beings have lots of genes, and what a gene is it's something that codes for a trait. It controls a trait. We'll talk about specifically how they do that in more detail next semester, and we'll see how these genes code for proteins and so forth. But for now, we'll leave that alone. So the genes that we have are, of course, in our DNA, and that DNA can exist in the chromatin state or in the condensed chromosome state. Now, as we've already looked at in this video, humans have 46 chromosomes total. That's how much DNA we have. But those 46 chromosomes exist in pairs. That's why we're diploid. And those pairs are called homologous pairs. Now, homo means the same. So what's the same about these chromosomes? Well, they are not necessarily letter for letter identical. They're not copies of each other. But they are homologous in that they are coding for, they have the same genes that are controlling the same trait. So let's pretend maybe, for example, this is a chromosome pair number 1, okay? And that we could have done any of them, right? 1 through 22 at least, or 23rd pair is a little different, but essentially the same. And so these pair then, we get one of those from our dad that came from the sperm and one that came from mom in the egg. And so now together we're diploid. And so what these chromosomes have is the same genes. So maybe in this region of the chromosome here, we have genes that are controlling eye color or hair color, right? Maybe we got, blonde hair from mom and, brownish hair from dad. And so we're going to be some combination of those traits. And we'll look at, how does that work with dominance or recessive and all that kind of stuff later on in this unit. But for now, understand that's what homologous pairs of chromosomes are. So when I look at my karyotype here, realize what I'm looking at. These are all homologous pairs of chromosomes. What that means, notice that the banding pattern looks the same. The banding pattern looks the same because the DNA here is very similar because it's coding for the same genes. Now, it might be different versions of those genes. Again, genes for blue eyes, genes for brown eyes, but it's the genes that control eye color. And so for each of these chromosome pairs, imagine we got one from mom, one from dad, one from dad, one from mom, one from dad, one from mom, one from mom, one from dad, and so forth, right? For each of these pairs. That's the way it works. These are now homologous pairs. Now remember, a gene is the actual segment of the DNA that actually controls a trait. How it does that, again, we'll look at later on. But for now, we're just going to leave it at that. Now, sister chromatids. Let's remember what sister chromatids are. What sister chromatids are, these are structures that only exist when the cell's dividing. And what happens is the mother cell, right? Remember, mother cell just means the first cell. So we're not talking about necessarily for mom or dad, right? The mother cell, after she's copied her DNA and she's getting ready to divide, she's going to condense her DNA into these chromosomes, and she's going to attach the exact copies together as a pair of sister chromatids. So when you look at a pair of sister chromatids here, what we're looking at, this is letter for letter actually identical. This is exactly the same. This DNA and the new copy of it are attached together. They are letter for letter the same. Same thing over here. These two chromosomes are identical to each other. Now, they are also still a homologous pair, right? We got one from mom, one from dad. But now they've been duplicated together to form these pair of sister chromatids. So we'd call this a pair of sister chromatids that's also now homologous. So if that lost you a little bit, let me kind of explain, right? So this is the initial situation. We have our homologous pair of chromosomes. Now in the S phase of interphase, of course, the mother cell copied all of this DNA. And she's now taken this chromosome with its exact identical copy and attached them together as a pair of sister chromatids. She's done the exact same thing with this one, form a pair of sister chromatids. So these are now each, instead of one chromosome, a pair of sister chromatids, but of course they're still homologous, just like they were up here. But now we're looking at them as a pair of sister chromatids versus individual chromosomes. So in a minute we're talking about meiosis, and we'll actually be drawing it. Try to keep that in mind, the connection between sister chromatids and homologous pairs. The nice thing for us is that if we understood the last unit, we understand how mitosis worked. Meiosis, the special kind of cell division, is not that much different. So let's remember, in mitosis, what happens is our somatic cells, so our non-reproductive cells, right, skin cells, liver cells, brain cells, whatever, when they divide, what happens is you have a 2N, a diploid mother cell, produces 2 daughter cells. And how do those daughter cells compare to the mom and to each other? They should be identical, right? So they should be also diploid, just like mom was, and they should have the exact same number and kind of chromosomes. They should be genetically identical to the mom and to each other. That's what mitosis is all about. It's about making cells that look the same. In meiosis, it's quite different in that we have one mother cell that is diploid, but she's going to divide essentially twice, and so as a result, instead of the daughter cells looking identical, the daughter cells are going to have half as much DNA as her. So she's going to make, and again, not 2 cells because she's going to divide twice, she's going to go from 1 to 2 and then from 2 to 4. So she's going to be making 4 daughter cells that are each going to be haploid, and they will not be genetically identical. Make a little note. They're not going to be identical to the mom cell, and they won't be identical to each other. They will be different from each other. Meiosis is the special kind of cell division that produces our gametes, which are the sperm and the egg again, so that depending on which organism or individual we're talking about, male or female, we're going to be producing either sperm or an egg. And this, in humans, and in all species that do sexual reproduction, meiosis reduces the chromosome number in half. Now, in humans, that's from 2N to N, right? From diploid to haploid. Again, in our, like, let's say strawberry plants are going from 8N, which is octoploid, to 4N, which is tetraploid. But in humans, we're going from 2N to N, and that way the sperm and egg will each have an N number of chromosomes. You put them together, and you get back to the 2N, the diploid zygote. And this happens actually by separating out those homologous chromosomes. So you're going to separate those homologous pairs. And what's nice about it, again, if you learned mitosis well, you know about prophase, metaphase, anaphase, telophase, and then cytokinesis, essentially it's going to be happening twice. So we're getting 2 sets of cell division, meiosis 1 and then meiosis 2. So it happens twice. We get 2 rounds of cell division, and that's going to produce not 2 daughter cells, but 4, and they're going to be genetically different. We're going to introduce what we call genetic variation. The fact that the sperm and egg that an individual produce can be very different from each other. Very similar to the steps of mitosis, but they're going to be happening twice. So, you have this on your study guide. You have interphase. So interphase, what happens in interphase? Well, I know this, the cell does what? It grows and it copies its DNA. Then it's going to go into prophase, metaphase, anaphase, telophase. Now we call it 1 because it's going to happen a second time. So this would be meiosis 1. Make a little note over here, of course, there is a cytokinesis. But then, instead of going back into interphase, instead we go right immediately into another round of cell division. Prophase 2, metaphase 2, anaphase 2, telophase 2, and again, of course, cytokinesis. So this is how it actually works. You're essentially going to be looking at this, and then these are going to be very similar, right? Very similar to what happened in mitosis. The chromosomes condensing, lining up, separating, 2 new nuclei. Chromosomes condensing, lining up, splitting, and then hopefully 4 new nuclei, and then 4 daughter cells. So this is going to be very similar to what we saw last unit in mitosis, but it's going to happen twice. We're going to have with 4 daughter cells, and they'll have exactly half as much DNA as the original mother cell. I'd like you to label this picture just the way it shows here. So here's our mother cell. It starts off as 2N. Let's skip this for now and jump down here. And what we're going to do is we're going to go through 1 round of cell division and then right into another round of cell division. So we have, instead of 2 daughter cells forming, a total of 4. And here, our daughter cells are going to be N. They're going to have just 1 chromosome for each pair. Now, the crossing over that's shown here, we'll look at that in more detail in a minute, but essentially what's happening is, notice these chromosomes, because they're homologous, they're very similar chromosomes, and so if you have them bump into each other, they can actually swap whole segments of chromosome, which is what they're showing here. They're kind of coming in together, bumping in together, and now you have a chromosome that's completely different to any chromosome that the mother cell has. It's this sort of mixed chromosome that's partially red with partially blue. We'll look at that in a little more detail in just a second. All right, right now what I want you to do is get that Chapter 11 window pane in front of you. You'll need a pen or pencil as well, and 2 colors. Any colors, I'll be using red and blue again. As you're drawing, make sure you're saving some room at the bottom here, and I'll be illustrating that in my window pane. So I'll be doing all my drawing up here, and then you'll be writing stuff in here. So it'll be very similar to our mitosis window pane that we did a little while ago. All right, so just so we're clear, whenever I'm writing with my green pen, that's going to be your regular pen or pencil. And then again, I'm going to use the colors blue and red. You can use whatever 2 colors you chose whenever we do that. Now, before we draw anything into box 1, up here at the very top, right up here, I want us to write, I'm going to do it over here, but I want you to write it way up here. Let's write the karyotype for the mother cell. And for this particular mother cell, we're going to have a simple organism that has just 2 pairs of homologous chromosomes. So again, that's your regular pen that I just wrote there. And we're going to have kind of a longer one and a shorter one. And we're going to be using red and blue. And the reason I'm using red and blue is kind of universally red or sort of pink is for girls and blue is for boys, right? Kind of that old school kind of thing there. And so what I'm trying to illustrate is here, this individual, it got 2 chromosomes from its mother and 2 chromosomes from its father. So if you want to label this, kind of maybe put a bracket right here and label it, this is the mother cell's karyotype. The other thing that I think you should write, and I think you should write this right where I'm going to write it, right on my window pane here, right to the side, is either put a big capital I for interphase or even write the whole word interphase right along here along the side, because we need to know the interphase, of course, is happening, but we're not going to show it. We know what happens in interphase. We know the cell grows and copies its DNA. It's the exact same interphase that we're used to seeing. Now let's actually look at drawing what's happening in our window pane. Our first window pane, what comes after interphase? It's going to be prophase. So we know what prophase looks like. It's going to be a circle. In that circle, we're going to have our centrioles that I'm going to draw as these kind of letter T's, because that's what they actually look like, right? The centrioles are producing a spindle apparatus. We know that the nucleus is breaking down. We'll illustrate that with our dotted line circle. And we're going to draw our chromosomes. Now, of course, each one of these chromosomes has condensed as a pair of sister chromatids. But now watch the way I'm going to draw them. My longer pair, what I'm going to do is I'm going to draw them doing a little bit of crossing over. So just my longer pair, not my shorter pair, I'm going to have them bumping in only right there. So they're going to be bumping into each other right there. I could have also had them kind of crossing over right here. I could have had them crossing over both of these little chromosomes. I could have, but I'm not going to. I'm just going to have just that little bit right there. My 2 smaller chromosomes are not going to be crossing over. They'll just be in the cell kind of like that. So this that I've just drawn, this is prophase 1. So in prophase 1, notice what happens here. The chromosomes can condense, or will condense. The nucleus appears, the central and spindle apparatus appears. This is all just exactly like prophase before. Highlight right here. This is the only new special thing. Crossing over can occur, and that's what crossing over is right there. So the only place that crossing over can occur is in prophase 1, and we drew just a little bit of it. We could have drawn more, but we didn't. We're going to keep it kind of simple. Moving on to our next box then. So from prophase, we know we get into metaphase, right? Now this metaphase is going to look just a little bit different than what we're used to seeing. So we've got our cell. We have our centrioles. Where am I drawing my centrioles? Of course, I'm drawing them at the poles, like we know. Now the thing that's a little bit different about this particular kind of metaphase, yes, the chromosomes are going to line up along the equator, but they're going to line up as a pair of homologous chromosomes. So watch what I mean here. So we've got our first pair, and let's go ahead and draw our second pair, a little shorter, right? Now notice what I'm doing here though. You see how now we've created this new chromosome that is blue and red and kind of red and blue right there? That is the result of our crossing over event that happened in prophase 1. So because those 2 chromosomes bumped into each other right there, they actually swapped whole segments of chromosomes. Because they're homologous, they're very similar. The genetic sequence is very similar. And so you can actually get swapping there. Now, why are they lined up along the equator? Again, it's not by magic. It's because of the activity of this spindle apparatus, just like in mitosis that we learned about. So very similar. This, again, is metaphase. This is metaphase 1. And it's the homologous chromosomes. Might want to underline or highlight that. The homologous chromosomes are lining up along the equator. Not the sister chromatids like we saw in mitosis, but the homologous pairs actually line up. And so if it's the homologous pairs that are lining up, they're going to be also what's separating in anaphase 1. So let's move on and let's draw anaphase 1. So anaphase 1, I'm going to draw, again, kind of more like an oval versus a circle. Draw my centrioles at the poles. And what I'm going to draw now is I'm going to draw my homologous pairs separating. So let me explain what that's going to look like here. So I've got my sister chromatids. They're still attached together as sister chromatids. All right, so I still have my sister chromatids. They're still attached, but they're being pulled apart. The homologous chromosomes are being pulled apart. The ones that were just lined up a minute ago. So they now are separating. And again, I have my first pair is longer than my second pair, so I'm drawing the second pair short, kind of smaller and shorter. And that, of course, is happening because of the activity of this spindle apparatus. So very similar to what happened in mitosis. So this, what we've just drawn, this is anaphase 1. Notice what it says. Again, you might want to highlight or circle or star, whatever. It's the homologous chromosomes that are separating, and they're pulled to the opposite poles where our two new daughter cells are going to form. So from anaphase 1, we're going to go into telophase 1. In telophase 1, we're going to draw our peanut. Just like before in mitosis, we're going to draw the centrioles. We're going to draw the new nuclei that's formed. And now realize the DNA does relax back into chromatin, but we're still going to draw it as chromosomes just so that we can trace the movement of our specific chromosomes. Okay? So this is going to be the one thing that's not going to really match up with what we're writing, is that we're going to still draw them as sister chromatids, even though in reality it does relax back into chromatin. And so there we go. And again, we can see the result now of our crossing over event. We have these separate chromosomes. They're a little different. And this right here, this is telophase 1. So again, we're not illustrating this part about the fact that the chromosomes are relaxing, but we're getting the two new nuclei forming. So apparatus breaks down now. Again, we're not going to show everything, so we're not going to draw cytokinesis. You can either write a big capital C here or write the word cytokinesis along the side. And then let's move right into our next box. Understand that there is no interphase. We're going to go right back into another round of prophase, but we have to keep track of the fact that now we have two cells that are doing it. So here's what's interesting about boxes 5, 6, 7, and 8. They're going to look exactly like the prophase, metaphase, anaphase, and telophase that we drew in mitosis. Exactly the same. The only difference is that it's going to be happening in two cells. So I've got my centrioles at the poles. I got the nucleus breaking down, spindle apparatus forming. And it's happening twice. And let's draw the condensed sister chromatids. And what we've drawn now is prophase, but this is now prophase 2. One easy way to be able to identify, because on the exam I will show you pictures, one very easy way to know if it's prophase, metaphase, anaphase, telophase 2 or 1, you should know what prophase, metaphase, anaphase, and telophase look like as far as what's happening. But how do I know if it's 1 or 2? Very simple. If I see 2 cells doing it, it's prophase 2. If I see 1 cell doing it, it's prophase 1. If I see 2 cells where the chromosomes aligned along the equator, well, I know that when they're aligned along the equator, that's metaphase, and so there's 2 cells doing it, it's metaphase 2. If there's 1 cell doing it, it's metaphase 1. So from prophase 2, we're going to go into metaphase 2. So again, we're going to draw our 2 cells. We're going to draw our centrioles at the poles. And we're going to draw now, again, this is going to look exactly like mitosis, so it's not going to be homologous pairs lining up. It's going to be our sister chromatids lining up, just like they did back in mitosis. So the sister chromatids line up along the equator. And of course, how they're able to do that is because of the activity of this spindle apparatus. What I've just drawn is, of course, metaphase, and this is happening in 2 cells. This is metaphase 2. So it's the sister chromatids. So you might want to kind of highlight that. You should know that in meiosis 1, it's all about separating homologous pairs. In meiosis 2, it's all about separating sister chromatids. Let's move on. From metaphase comes, of course, anaphase. For anaphase, I'm going to draw 2 ovals. Draw my centrioles at the poles. And I'm going to now have my sister chromatids separating. Again, this should look very similar to mitosis. And the reason why they're separating, of course, is the activity of that spindle apparatus. So spindle fibers shortening, controlled by the centrioles. We're talking about an animal cell. And that, then, is anaphase 2. So in anaphase 2, it's the sister chromatids. Again, I want to highlight whatever realize it's the sister chromatids that are separating in meiosis 2. Let's move on now to our last box. In our last box, we're going to draw telophase 2. So we're going to draw 2 peanuts. Draw our centrioles at the poles. Draw our now 4 nuclei that are appearing. Now again, the DNA does relax back into chromatin, but I'm going to still draw it as chromosomes just so we can see how everything kind of ended up. What I've just drawn is telophase 2. So realize with telophase 2, the chromosomes, they do relax back into chromatin, though I haven't drawn that. You get 4 new nuclei are forming, and the spindle apparatus breaks down. And these then will go through again. We should label cytokinesis, of course, is going to be happening. We do that in your green, cytokinesis. And if you think about the daughter cells that we've created, if you compare them to our mother cell's karyotype, for chromosome pair number 1, the daughter cells will get just 1 chromosome. For chromosome pair number 2, they're going to get just 1 chromosome. And notice also that we have 2 of these gametes are going to get a chromosome. If you think about it, if you want to make a little note, whatever, because of crossing over, if you think about it, if you compare these chromosomes to what the mother cells has, what you'll see is that these chromosomes are actually not found in the mother cell. These are brand new chromosomes, a new mix of DNA that the daughter cell has. That the mother cell actually doesn't have that exact same combination of genes. So again, if you want to summarize that idea in your own words, you can just say essentially, right, that these 2 daughter cells, because crossing over happened, they're going to end up with a chromosome that is unique to them. It's not found in the mother cell. That's going to be a source of genetic variation in the next generation. All right, so one of the many reasons why you're different than your parents is because you get a mix of their DNA, and you can actually get mixes of the DNA that maybe each of them individually don't have. So pretty cool stuff. That is the end of that video on meiosis and kind of setting up sound foundation. Hope that was very helpful, and I'll see you next time.